Qualcosa è successo.
Sì.
Perché ho avuto l'annuncio, quindi...
Esatto, adesso vediamo se mi rindirizza tutto. Ok, we are live.
Ok.
Nella mia condivisione dello schermo, diciamo, i volti, le telecamere, inclusa la mia, nel mio schermo sono in alto a sinistra. Li vedete anche voi? Devo minimizzarli, per esempio, così, perché magari danno noia, come... Non sono sicuro che sia... Adesso li ho espansi, non sono sicuro che...
Io vedo solo la tua prima slide.
Allora, perfetto.
Quella col titolo affiliazione.
Perfetto.
E basta, quindi Zoom offre sempre il cambio di software tutte le volte, richiede uno sforzo. Ok.
Buongiorno, lo speaker si sente a volume abbastanza basso, non so se si può alzare un poco il livello.
È un po' meglio così? L'importante è che non saturi.
Molto meglio. Molto meglio.
Ok, chiedo scusa. Ok. Ecco qua.
Ok. Oggi so che c'è poca gente oggi, eh? Vogliamo comunque partire e poi magari si aggiunge qualcuno? So, yeah. So I switched to English as clearly the talk is in English. And so it's my great pleasure to have today Michele Giuliano from CISA in Trieste. So Michele graduated and got a PhD in Italy, and he moved for a postdoc in Switzerland, and progressively he went more to the north, let's say. So he arrived in Antwerp, where he got a position as an assistant professor. And since some years, so it would say 2017, he's 19 actually. He's in Italy at the CISA of Trieste. So Michele combines nice neuroengineering approaches with also, I would say, computational approaches. And I think you will clearly appreciate that from his talk. And he will deal with correlation, which is quite an important subject in biological tissues. And I think his approach will nicely translate what we know from experiments to the theory. And so I think the best is if I leave the floor to Michele directly. And for questions, as we discussed before, you can interrupt at any moment Michele, or wait till the end for the questions. So Michele is prepared for any possibility. So thanks, Michele, and also Marco for co-organizing the seminar.
Okay, so thank you, Thierry, for the introduction and for the invitation. It's an honor to be here at this virtual seminar series. You will see that in this talk, I'm going to roughly combine two domains. One is purely neurobiological, and the other one is sort of inspired and motivated by theory, by computational neuroscience. And the main character of the talk is correlations. I will try to give some insight. What do I mean with correlations in the context of the brain? Which is the level and which is the exact definition that I'm going to use to tell you about some experimental results that we got. So indeed, I am in, well, right now I'm not in Trieste, but I come from CISA, where I have my lab. And the lab is combining three disciplines, all aimed at the overall goal of understanding or characterizing the biophysics correlate of computation in the brain. And for the talk of today, there are these two disciplines. So one is physiology, electrophysiology of cells and circuits. In today's talk is about cells, and modeling. So we're not talking about any neurotechnological tool to support the investigation of the activity of cell circuits and in silico, air, and in vitro. So I start from the end, and I will acknowledge, I'm acknowledging particularly the support and the work of my current lab members, as well as of a previous postdoc who is currently assistant professor at the Politecnico di Milano, Daniele Linaro, who actually did the work, did the experiments that I'm going to tell you about. And this work has been done in collaboration with Brent Doiron at the University of Chicago and Gabriel Oker, who is now in Boston and previously was at the Allen Institute in Seattle. So this is kind of the menu. Let me minimize, and yeah, I minimize the cameras. So this is the plan. I'm going to tell you about the background of correlated activity and the consequences, giving you ultimately a definition. And then I will tackle the question, which is, how is input-output correlation transfer in a specific type or class of cells from the cortex of rodents? And I'm going to tell you that indeed there is diversity in the way that correlations is occurring, taking place, are transferred from the input to the output, depending if the neuron is excitatory and inhibitory, although it's more subtle than that. And I will also tell you about different global brain states and how these are able to influence the way that this correlation transfer is taking place. This is conventionally called in the literature high conductance states and low conductance states. And I will then speculate about possible functional implication. Ultimately, depending on the time, I will conclude giving a bit of highlights on one specific theory, which is very dear to an engineer. By training, I'm an electronic engineer. And so the linear response theory is key, and it could be validated in the context of these experiments. So first of all, the correlation, the way I'm going to use it in this talk, is a measure of covariation in a cell pair. So I'm actually focusing at the cell level, thinking of pairs of cells, no matter how far they are, no matter if they are embedded in the same microcircuit or different brain areas. And I'm actually referring to what in the literature is called and known as noise correlation. So I'm not talking about the so-called signal correlation, which is reflected in the so-called tuning similarity. You probably know, say, for instance, in the visual cortex, there are cells that are sensitive to the orientation of a stimulus, and maybe neighboring cells might have some shared similarity of their tuning preference. So if you present a stimulus, then both cells will actually have a sort of correlated activity. This is not what I mean. In this talk, I'm focusing on the other possible definition, which is the degree by which trial-to-trial fluctuations in the response of these pairs of neurons are shared. So it's stimulus independent in a way. And it has been reported through a variety of techniques, ranging from 2-photon, looking at concentration of calcium, stabbing cells in the stomach and looking with a pipette by means of electrophysiology, which is going to be actually the technique of today, looking at their intracellular potential, the membrane potential that is somehow correlated in neighboring cells. You actually see here the black and the blue trace, as well as at the level of the extracellular, the population, the ensemble activity in terms of spikes detected even in vivo during awake and behaving tasks. And it's not only in the cortex, which is by choice the theme, the context, and the preparation that we are using in the lab, but it has been described that even in the retina, in the periphery, you actually have correlated, covariations, which are reported in the spontaneous firing of retinal ganglion cells, but also in the LGN, which is in the visual system. It's another station. It's a station going towards the cortex, as well as in other sensory thalamic areas, such as the somatosensory thalamus, ultimately individual cortex. But by no means these are only featuring sensory cortices. So yeah, somatosensory is involved, or has been one of the place of the where correlations has been described extensively over the past 20, 30 years, but also in the motor areas, as well as in the associative areas, for instance, the IT, inferotemporal cortex, or the PFC, the prefrontal cortex, those areas of the brain which are usually associated to associations, to decision-making, say, task and computation, which are less obvious than the purely sensing and perception. These are three examples, and I'm actually coming, I'm reporting some three very pioneering studies from the output to the input of pairs of cells. So looking at the outputs, what you actually see at the top are 4 so-called spike-triggered cross-correlograms. So in a matter of a time window, in this case of 200 milliseconds, they are reporting the excess probability of firing of cell B, given that cell A has just fired. So if you actually see here these horizontal axis 0 or plus 200 or minus 200, it refers to the lag compared to the every nerve impulse fired by cell A. And what you actually see here is a recording in an awake animals, an awake non-human primate, which has been receiving auditory stimulation. And these 2 cells are showing basically no correlated activity. You actually see the blue is sort of flat, so there is no excess probability of firing. If you want, trivializing, no synchronization, although it's a little bit more complicated than that, before and after the presentation of the stimulus. But during the presentation of the stimulus, the activity of these outputs, these trains of spikes, are showing some correlated activity. And this is over this time scale of tens or hundreds of milliseconds. Moving towards the what is generating the action potential, the nerve impulse, here I'm showing 2 traces from another very complex and pioneering studies in the mouse by a colleague, Luc Gentet, who in vivo, in a awake, had restrained mouse preparation. He could patch, or in other words, he could insert 2 glass pipettes into the stomach of 2 cells in the somatosensory cortex of a mouse, and those 2 cells were both excitatory. And there are molecular ways to actually guide this otherwise blind patch clamp experiments. And you actually see that what he recorded is the constant ongoing evolution through time of the electrical potential, electrostatic potential, the transmembrane potential is also called. And in these 2 cells, which were probably apart from a few hundreds of micrometers, there are very strong and very high similar deflections and fluctuations. So even at the level of membrane potential, not only at the input, at the output, when the spikes are generated, even before, when the integration takes place, neurons show strong similarity. And this is also the case at the level of synaptic input. So going at the bottom of the slide, the technique is a little bit tricky. I don't enter into the details. There is a way by the same 2 pipettes in the stomach of a cell to electrophysiologically isolate what is an excitatory input and what is an inhibitory input. And in these 2 cells, on the left is excitatory and on the right is inhibitory. Blue and red are 2 cells, and you actually see that they are receiving a bombardment of synaptic inputs, which is highly similar. And the time scale here is similar. I apologize, here there is no calibration bar. This is probably of the order of 10 to 100 to 100 milliseconds. Now, people have been very well aware of this, of the presence of correlation. And since the 90s, where other very key experiments on primates have been carried out, they look at this as a sort of a bug, not of a feature, because for information encoding, if you have redundancy, okay, maybe it's a resistance to noise, but you are not doing good, especially if you have, say, 1,000 neurons or 100,000 or 1 million neurons. The more you increase the number of neurons, you don't necessarily increase the content of the information content if the neurons are not independent in a statistical sense. What you actually see here is the formula. If there is interest, I will go through it. It's just a cartoon to demonstrate that if you have m random variables, which are, say, Gaussian distributed, and they are, however, correlated, so there is a correlation coefficient R between any two pairs of this set. And if you conventionally define the signal-to-noise ratio as the mean divided by the standard deviation, you find that there is the presence of a correlation. So if R that there is at the denominator is non-zero, then the more you increase the number of these variables, m, the less you are going to improve the signal-to-noise ratio, which is by no means linear. It's only linear for R equals 0 up to roughly 100 for this choice of parameters, because otherwise correlations are bending this signal-to-noise ratio. So you can fix the signal-to-noise ratio that you're happy with, and even if you observe more and more and more neurons, you don't gain that much. So this has been well known as a problem, as a bug. Other people, other investigators in the context of decision-making and optimal discrimination thought that the presence of positive or negative correlations could actually be advantageous for separating, like in this case, the a posteriori, so downstream, the stimulus that was in the environment, that was, for instance, observed by looking at the response of two cells, neuron 1 and neuron 2, that were both selective to stimulus S1 and stimulus S2. As soon as you introduce correlations, and if the correlations are in the right directions, those sort of Gaussian profiles that would be otherwise sort of circular, symmetric, they become more like an ellipse. So if you are after the definition of a boundary for making a maximum likelihood decision or discrimination, then this is going to be in your advantage. So this could actually be a feature and not a bug. And ultimately, this is what the intuition would tell. The more you have correlations, and here I'm using this intuition, which is by no means complete. The more the inputs to a cell are correlated, are kind of synchronized, the more spikes you might have, because you might kind of roughly exceed this excitability threshold that neurons have in order to emit an impulse, an action potential. So what has been shown more than 20 years ago by Salinas and Sejnowski, prominent theoreticians in the field, is that the Fi curve, which is a sort of Rosetta Stone of how neurons, the transfer function, the response function of individual cells, can be boosted in terms of the slope of this relationship, which is input injected and output rate of emission of impulses is actually boosted. It increases its slope the more you have synchronization of or correlation. So this might be a way that the brain used to increase the gain. And you probably know that gain modulation is one of the key computational primitives that are implemented in the brain at many different levels. And this is a sort of elementary biophysics level that involves the local circuit. I'm not telling you about in the context of pathological rhythms, like in this case from an animal model of absence seizure, that you have a pathological correlated activity, a pathological correlation between neurons. So in this talk, I'm focusing on one or two definitions of correlation. Specifically, I'm focusing on spike counts, and I'm going to tell you what I mean with spike count, and I'm looking at the covariance or the correlation coefficient. So imagine I have a bunch of neurons. I can observe simultaneously or not simultaneously, that's another story, but let's assume simultaneously 2 cells at a time. I can once more stab, I can put a pipette in their stomach so that I can maybe observe their spikes, the spike trains. There are those very rapid deflections of about 100 millivolts that are detected intracellularly. And I can fix a time window, say 40 milliseconds or 100 milliseconds or 1000 milliseconds. And in this time window that can be sliding or not sliding, I can count how many spikes I have. And so in this case, I have 1, 2, 3, 4, 5. In this case, 1, 2, 3, 6. I move this time window, and I end up with two time series of numbers, 5, 5, 7, blah, blah, and 6, 6, 8, blah, blah. And those are basically a realization of random variables. And I can look at the definition of covariance, which is the expected value of the product, well, okay, without the offset of the mean, and the correlation coefficient as a sort of normalized version of the covariance. This is key for consideration for an experimental findings that will come in a moment. So this is the way we operatively defined correlations, focusing on the spike count. And you see that by changing the time window from maybe 40 milliseconds to 5 milliseconds, maybe we can still capture this similarity or covariability at the level of what I just said, or I said before, synchronization, maybe that kind of sort of, maybe not sub-milliseconds, but roughly around the milliseconds of co-occurrence of action potential. So having defined this measure, the scientific question that we tackled are here, laid out here. Specifically, if I have correlated inputs, how are these inputs mapped in the output? Are neurons devices that tend to decorrelate their inputs, or they are maybe promoting correlations? How they are progressing or transferring this correlation from the input and the output? And if they do differently, is there any role for cellular or subcellular mechanism? Particularly, cells are known to be diverse, although in the mind of a physicist and of an engineer like me, we tend to get or to look for the stereotypical neuron, but they might show quite significant diversity in their electrical activity. So is there any consequence for that? And another question is, what theory do we need in order to look or predict this correlation and test it? I open a very brief parenthesis. The analysis of correlation transfer is key for some theoretical concept, which is somehow augmenting some theoretical tools, which is in computational neuroscience has been very successful over the past 30, 40 years, which is known as the mean field approximation to the activity of a network of neurons. If you want to go a little bit beyond the so-called sparse connectivity, then you have to understand and to add into your equation the correlation terms. So in a way here, we are interested in going back to the experiments and see how these correlations are taking place, how they are developing. And because we have experience and we love the advantage of in vitro brain preparations, so we actually do slices, very thin slices from the brain, specifically from the somatosensory cortex of a rat, we actually choose to focus on a very specific cortical microcircuit. And we look with a technique that I will comment on it in a moment. We look at so-called layer 5, cortical layer 5. The layer 5 is important because the large pyramidal cells that are here sketched on the left are known or considered to be the output from the cortex. So in a way, as an engineer would be obsessed with black boxes that I don't understand, the best thing would be, let me look at the output. What is this piece of cortex is communicating to other parts of the cortex or to downstream other parts of the brain? And at the same time, pyramidal cells, which are excitatory or glutamatergic, as it says in jargon, we also look at two other cell types that are inhibitory interneurons. One is called basket cells or PV positive. I'm not entering into the details. It's a molecular marker that would allow these cells to actually blink. So one could actually recognize them not only from their diversity of electrical response, but also from their color or their fluorescence. So PV positive, they are basket cells, and somatostatin positive, which are normally called Martinotti cells. Both are GABAergic interneurons, so they are inhibitory cells, but they have different firing patterns, so they are not the same. And the second ingredient that we use is an experimental technique that is called dynamic clamp, which involves a real-time operating system that is controlling an amplifier in real time, so that if we want to inject, and it will be clear in a moment, if we want to be the persons in charge of manipulating the input to these cells, we don't want to inject a current into the cell as a sort of additive input. Once more, trying to use the terminology that is maybe familiar with an engineer and physicist, because this current injection may not be accurate. Neurons are not communicating by exchanging currents, but rather by activating conductances. So the proper way is to have a current which is instantaneously depending on the electrical potential of the target cell. If the target cell is changing and you adapt the current in real time, what you're effectively doing, you're injecting by the Ohm's law, you're injecting a time-varying conductance or resistance the way you want. And this is known to be a sort of golden standard for increasing realism in driving neurons. And it's not just an academic curiosity. I'm going to report that if you don't do this, then you get different results, which is actually quite important. I'm not telling you about some technical details such as the active electrode compensation technique. So these three classes of neurons are well known for their diversity of electrical phenotype. And I'm not entering into the details. What you actually see here is that these three cells were patched, I mean one pipe that has been inserted in their stomach, and roughly the same amount of, in this case, it's a DC constant current. It's not a conductance, but this is a kind of sort of test to actually see the phase, the phenotype, the electrical response of these cells, to discriminate between them. They are responding in a different way. The pyramidal cells, the excitatory cells, they are responding relatively with low rate. You actually, even if you don't squint your eyes, you actually see that there are vertical lines, and these vertical lines, they show a little bit of adaptation. The first two spikes are very close, and then they start to slightly relax. This is called accommodation or spike frequency adaptation. It's an example of negative feedback. You're going too fast, and I'm going to slow you down. As you can see, instead, the inhibitory cells, the PV positive and somatostatin positive cells, they are both interneurons, but they're very different. They are called fast spikers, and you understand why they are called fast spikers, because they have a lot of spikes in the unit of time. And one of them is basically like a pacemaker. It's actually spiking without any relaxation or adaptation. Instead, the orange cell, so the Martinotti cell, you actually see here, you can squint your eye, you will see that it's dark at the beginning and then is relaxing, is instead accommodating, is instead slowing a little bit down. But they fire with a much higher number of nerve impulse per second. So they are diverse, and they have other properties that I'm not going to comment, but I can show you the recordings later if you want. Next, we had to find a way to control the input correlation. I said that being the experimentalist, we wanted to be in charge of completely controlling what would be in, say, two cells at a time, like these red and purple cells, what would be the fraction of correlated inputs that these two guys are sharing. And this is possible by a very simple protocol suggested many years ago by De La Rocha and colleagues, which involves assuming that cell number 1 and cell number 2 receive a superposition of two terms, one each independent, so the red is statistically independent from the blue, and one that is instead shared. And these, respectively, red and black and blue, are superimposed linearly. We can discuss about the accuracy of these of this assumption, and they are feeding by this dynamic lamp, by this conductance injection, real-time manipulation of the injected current, leading to a train of action potentials. Being the train of action potential, the choice of the time scale would allow us to calculate the covariance or the correlation coefficient. And because it's us controlling these red and black and blue inputs, we can control the parameters of these inputs that, strictly speaking, they are all realization of a stochastic process known as the Ornstein-Uhlenbeck process. I can tell you later why this is considered to be widely used and considered to be a sort of biologically realistic kind of input for cells. And we can therefore control many things. We can change the parameters in a very trivial way, mean invariance of these inputs to make the cells firing more or say faster or slower. And definitely by weighting differently the red independent and the common terms by this coefficient C, imagine that C is 0, then this branch will not be there. It's going to be only the red one. So red and blue will be the only drive to cell 1 and cell 2. Instead, if C is equals to 1, both cells will not receive any red and not receive any blue. They will all receive the same black. So we can, just by this knob, we can control the amount of correlated inputs. And this is somehow our control parameter. You actually see here that by changing this C from 0.2 to 0.8 and plotting, superimpose the spikes collected from the cell number 1 and cell number 2 in this experiment, black and yellow, you actually see that for 0.8, say 80% shared inputs, as expected, you have many more co-occurrence. Not always, but you have many more co-occurrence of action potential fired roughly at the same moment. So first result is that, as expected, output correlations are depending monotonically on the input correlations. And if you're an excitatory or inhibitory cells, doesn't matter. So this seems to be disappointing because it seems, wait, but then there is no diversity. It comes a little bit later when investigated at the more, say, in a slightly more, in a deeper way. What it's interesting to notice here is that there is no, so all these points are not lying on the unitary slope line because the cells in the pairs that I described to you are not identical. Any two neurons in the brain are not identical. So even if they are sharing 100% of the same inputs, they are not going to behave in the same way. So you have seen it before with a C equals 0.8, so 80% shared similarity. They don't do the same thing. Sometimes one spikes, the other one does not. But the key point, the key observation here is that if you look at correlations which are relatively large, say from 0 to 0.5, maybe 0.6, you have linearity. So the output correlation coefficient is linear, is proportional to the input correlation of the analog waveforms that we've been injecting in those two neurons. And this is key for the application of this linear response theory and the way that the correlations can be mapped in the output and how one can do this analytically to make predictions and to test these predictions. So this was one unexpected first result. Second result, which is, this one is highly counterintuitive, is the following. Say that I fix the correlation, say 2.5. So it's actually quite a lot, but I know that this behavior is still in the linear regime. And I also fix the time window, say 40 milliseconds. What I can do, I can change the properties, the statistical properties of the input, so that the two cells are firing with different firing frequencies, from very slow to very fast. Very fast means 20, 25 spikes per second. Now, I have a pair of cells, and the two cells are never going to fire exactly at the same value, although we tried to do that. So what we've been indicating here on the x-axis is the geometric mean firing rate. Is a purely conventional way to lump in one number what would be otherwise two numbers, firing rate of cell number 1 and firing rate of cell number 2. I'm just having one control observable, right? Control variable. And what you notice is highly unexpected. The covariance seems to increase the more spikes the neurons are emitting. And believe me, this has nothing to do with the false intuition that you might have saying, oh yeah, of course, you have more spikes. So you have more opportunity for these spikes to be correlated or say synchronous or non-synchronous. No, these covariance measures are normalized by the firing frequency. The offset is removed. So this is a sort of consequence of some nonlinearity, which we actually afterwards realized that has to do with the threshold linear nonlinearity, the existence of a minimal input amplitude below which you actually don't have any output. This is the reason why the larger the firing frequency, the higher the covariance. But that's not all. So they increase with rate And if you are an inhibitory cells, you actually vary even more or less 5 times more than the excitatory cells. But so basket cells are propagating, are transferring almost 4 or 5 times more covariance from their input to their outputs, whereas the Martinotti cells are slightly in between. So pyramidal cell seems to be not particularly interested in this, in the correlation in the inputs. I cannot say that they are decorrelating input, but they actually seems to not to promote this. Inhibitory cells seems to actually do the opposite. They favor, as soon as you have a little bit of correlation like 0.5, they actually tend to have, even the higher the frequency, the more correlation in the output. Do they want to act as a global quantity, as a global entity, whereas in excitation, that I will be speculating later on. Now, if I want to give a sort of take-home message, what I can do, I can take the data from the previous plots, and I can make a sort of ratio. So covariance of the basket cell divided by the covariance of the pyramidal cells. And on the right, covariance of the Martinotti cell divided by the covariance of the pyramidal cell. So that if it's 1, it means they basically have the same. And if it's above or below 1, I can say, well, 1 is better than the other. Better in the context that I'm telling you. Because this allows me to look at what happens by changing the firing frequency the way I did before, but also here changing the window T. And so here we didn't do any new experiments. We simply reanalyzed the data with a different T, time window for the definition of the spike count. And to make a long story short, for long time scales, you actually see here above 40 milliseconds, for both of these plots, all these points are above 1. It means inhibitory are winning over pyramidal cells. They transfer more, and they even transfer even more the higher the firing frequency. So here, if you look at the diamonds, in both cases, we're talking about higher frequency of the order of 15 to 20 spikes per second. So you're an inhibitory neurons, you're better than pyramidal cells, particularly at long time scales and high firing frequencies. Long time scales does not mean sub-millisecond synchronization of the actual spike times. This is the interesting thing. Spike counts, so it's a kind of global, slow signal that is correlated and it's passed from the input to the output, not the sub-milliseconds individual action potential synchronization. Let me actually see, maybe I'm going to skip this part where I was looking at this global brain state known as high conductance versus low conductance states that we could simulate because we were in control of this real-time loop for injecting a conductance or switching it off and injecting a current. But let me skip it and speculate about the functional implications. So what I just said, it seems from the data that pyramidal cells are operating in a regime where they are promoting low correlation levels. Even if there are input correlation, they tend to suppress them. Maybe they want to do that because they are aware of this redundancy problem that I indicated at the beginning. If you have correlations, then you are not favoring information transfer. So maybe they want to maximize information coding performance by reducing as much as possible the correlations. They don't want to be redundant. On the other hand, it seems that both basket cells and the Martinotti cells seems to efficiently transfer shared inputs in their outputs. And this is well known, or it's a sort of dear concept to the theoreticians or to people doing computational neuroscience and doing simulations, because inhibition in many, many models is called or defined as global. So you might have different subpopulation of excitatory cells with different recurrent and different patterns of connectivity, but then there is usually a single blob of inhibitory cells that are receiving excitation from everywhere, and then they're inhibiting everyone else. So this is known also with the term of blanket inhibition as a sort of, as a blanket that is laying down on all the neurons from a network. And has been by Raphael Yuste and collaborators in this review from 2015, has been also somehow linked to some temporal scales, because these Martinotti or basket cells, they are known to have GABAergic synapses, which are themselves time varying. And so it might be that they act on a different time scales. So here I'm hinting at concept known as short-term facilitation or short-term depression, also known as almost synaptic transmission or dynamic synaptic transmission. So it can be linked to also these other two things. The third elements that I'm going to speculate about is the so-called pyramidal interneuron gamma frequency oscillation, gamma rhythm generation. It's very trivial if you think of it, that you might have rhythmic activity if you have a sort of ping pong between excitation and inhibition. Actually, ping is an acronym for pyramidal interneurons, gamma, but it's also some sort of pun for having ping pong between excitation and inhibition. The excitatory neurons are exciting the inhibitory neurons, but once the inhibitory neurons are excited, they are inhibiting the excitatory neurons, which are basically becoming silent. And if they become silent, they are not any longer exciting the inhibitory cells, which are not anymore inhibiting the excitatory neurons. It's difficult to say it aloud correctly, but it should have given you the impression of a sort of alternation, which is indeed known at time scales which are relevant to EEG rhythms, particularly during gamma range, during specific frequency of activation of cortical as well as hippocampal circuits, to be associated with tasks such as cognition or locomotion or declarative memory. So parvalbumin positive, so basket cells particularly, seems to be crucial for the generation of this rhythm, because they are specifically impinging or projecting onto pyramidal cells in a very strong and effective way. That this was roughly hinted at the very beginning when I had the sketch where different interneuron would project normally, and this is not something that we tested in this experiment, in different parts of the morphology of a cell. So very quickly, if you're fond of mechanistic interpretation, you can look at some sort of mathematical models that does not need to be ultra complicated. These are a family of models called integrate and fire. I don't know whether any of you has some familiarity. They are spiking neural models. They conserve by physical realism. And in the specific case, they are known as exponential integrate and fire. There is something about the initiation of the action potential, which is a little bit more sophisticated than the kind of vanilla integrate and fire. They are called leaky integrate and fire. If you do things properly, combining integrate and fire and accommodation or spike frequency adaptation with some sort of variable that is mimicking the accumulation of a certain ionic species, which is providing a negative feedback, you can replicate qualitatively the difference between the experiments, both at the level of these transfer of correlation as a function of the frequency. Remember, the blue ones are transferring 5 times more than the black, the pyramidal, so baskets are winning over the pyramidal cells. The somatostatin positive or Martinotti are in between, and this is something that I did not tell you about, are related to this global state. I'm not entering into the details, but the story that or the tools, the theoretical tools that I described, that I hinted at the beginning, saying the linear response theory, is the ingredient that allow us to test whether the results were aligned to our prediction. And it's in the best tradition of engineering. Indeed, they are looking at individual neurons as a black box and characterizing both in time or in the Fourier domain the response of a neuron to a stimulus as a filtering operation, basically. In the Fourier domain, this H of omega is called transfer function. And our lab was the first to characterize these transfer function across a variety of cells, including human pyramidal cells, finding that human cells have a broader bandwidth. That would be another story. And so this filtering and the details of magnitude and phase, yes, I'm an engineer and I'm obsessed with Fourier and the magnitude and phase description of linear systems. These two ingredients can be mathematically applied to pen and paper, calculate what would be the transfer of correlated activity in the input to correlated activity in the output. I don't tell you how this is done. If you're interested, I will go later on. If there are questions, I will tell you later how this is done. It's not particularly complicated, and allow us to make a very simple prediction for a specific choice for approximating those integral that you have seen before, those anti-transform. So for a specific choice of this time window, which is relatively large, 40 milliseconds, we had a sort of formula. The covariance should behave as a sort of product of the transfer function in the Fourier domain at frequency equals to 0. So some of the feature of those filter properties. And what you actually see here in this plot are the markers, are the measure covariance, and sorry, apologies. So yeah, they are the experiments, and on the vertical axis you have the measured, and on the horizontal axis you have what is predicted. And indeed, they are aligning on the line, which is the coefficient of proportionality that we could not, that is the free parameter that we could not estimate for simplicity. This works if you have a pair of pyramidal, pair of basket cells, or pair of somatostatin cells, as well as in the reanalysis of the data, we could do all the possible combinations. It works also if you have mixed terms, pyramidal and somatostatin positive, or basket cells and basket and Martinotti cells, which is the demonstration. So summing up, I apologize for the lack of time for the story of the high conductance states and low conductance states, but I hope that in a way, in an accessible way, I convince you, or I reported you that the correlation transfer has some unexpected feature. It depends on cell type, especially for long time windows of spike counts and high firing frequencies, with the clear winner, which is one subtype of inhibitory interneurons, the so-called basket cells, or PV positive, parvalbumin positive cells, which are able to transfer about 5 times more covariance than the pyramidal cells. The other type of inhibitory interneurons is actually in between, and it's kind of defend itself in a decent way. It transfers less. And you don't need highly complicated so-called conductance-based Hodgkin-Huxley-like or Human Brain Project-like morphologically detailed models, mathematical models and simulations to predict what is happening. And the linear response theory that is per se would be an interesting theme to talk about, revealed to match very well the experiments, and then going back, suggesting that this description in the way that you can understand how dynamically individual neurons are transferring input into the output is a good one. Because if you look in the context of correlations, you can actually interpret quantitatively the experiments. I think I will stop here, thanking you for your attention and taking questions. Thank you.
Okay. Thank you, Michele. So it was a very good talk. Thank you for going through this research. It's actually connected to a paper that you, I think you mentioned in the slide. So are there any questions from the audience? Otherwise, I can start. And well, I was wondering if maybe you said it, but for the excitatory neurons, actually the lower performance is connected to also to the adaptation, or is that just by accident?
It was our idea at the beginning, because immediately after one action potential, if you have an adaptation, you have some sort of negative feedback. And that's the reason why, let me see if I can go to the slide that I want. That's why we look at the somatostatin positive or Martinotti cells, because they do adapt, and didn't seems to be the case. What turns out to be the determinant, let me actually go. Okay, so apologies, I don't want to cause any seizure, but so also the somatostatin positive are adapting. No. And they do fire normally at the higher firing frequencies, but in the way that we adjusted the inputs, we basically make all the cells firing in the same firing frequency range. So yeah, it could be that cells would have fired differently, but no, we made them firing in the same range. So it cannot be that. What it seems to play a major role, at least for very long windows of integration, is the slope of the Fi curve. So ultimately, the fact that the fast spikers are starting with a bigger slope, despite they are brought to a different regime, is the key for understanding or changing, at least in the limit of big over long time for the spike count, to interpret this diversity. So there is a range between pyramidal, Martinotti, and basket cells, and this is indeed reflected in this performance. Pyramidal are the worst, or take the worst in transferring correlation, then Martinotti, and then basket cell.
Right. And actually, I have another question related to, let's say, the debate between spike count and time coding. Let's call it this way. So since I guess many cells are transferring information actually in a very short time window. I mean, how could you somehow, say that with long time windows, these cells are really doing something out of these inputs? Because I would expect that, well, at short time windows, it's really important what the cell is doing. At a longer time scale, probably yes, but I'm less aware of what the cell might do on a longer time scale.
The way that I explain to myself, which is clearly only a partial explanation, I don't have any proof, is that these long time window spike counts is somehow reflecting or giving a sort of ground of the overall network activity. So I will push it provocatively. It would be as if spikes are not really, they don't matter. Units could be firing rate models, where you don't have spikes, but you have an analog continuous variable that is telling what the neuron is doing in the output. So therefore, inhibitor neurons are actually tend to shut down at that time scales the excitatory cells behaving as these blanket inhibition, because they have to set up the dynamical point. What I didn't tell you, what I didn't present because of time, which was this high conductance versus low conductance. Let me only look at the conclusion. So by doing the same thing, so I had indeed two conditions. One that we think it's similar to high conductance, where you have conductance clamp, dynamic clamp, and one that is basically purely current injection. And we call that the latter low conductance. So we did basically this ratio between the covariants, so we could see what was winning above 1 and what was losing below 1. So we could conclude that high conductance states are better transferring short time scales covariation. So if you are having a very strong, intense bombardment and thinking of the way that Alain Destex and others and collaborators were the first defining or studying, even in simulation and experimental, these high conductance states, well, in high conductance states, a membrane time constant and the slope of the Fi curve are changing appropriately. And it seems that the way that there is a highest transfer of correlation is precisely on a lower window of the spike count, thereby individual spikes, synchrony. Instead, low conductance states, and I'm not, although it was in the slide, but I'm very careful, I could maybe compare low conductance states with anesthetized conditions. So not a wakefulness that would be similar to high conductance. In anesthetized conditions, low conductance in this protocol in vitro in a dish, so we didn't have any sleeping preparation or any anesthetized preparation. It was a piece of brain in a dish in vitro. But in this case, you have the opposite. They are better transferring long time scale covariation. So this information content, whether it is on a very short time scales or a long time scales, it may be having in terms of correlation transfer, may have a different fate depending of the state of the brain of the network, if the network is awake or the network is not awake. This is clearly a speculation because maybe I can be criticized very easily for linking high conductance states to wakefulness and low conductance states with sleep. This may not be the case. Also, up and down states during sleep would make this very trivial and naive interpretation of mine weak.
Okay, thanks a lot. Hope there are other questions in the audience today.
Yes, hi. Ciao Michele and Giovanni. Ciao. So, no, sorry, I apologize because I missed the first 5 minutes, so I don't know. Okay, I have also curiosity. I mean, the first things is about, do you think if your approach, which is typically approach is using transfer function, is possible to, in some sense, to study also some pathological things, or so as some hypothesis about how the wall circuits, the transfer of information over circuits is in some sense in some pathological state. So I have some hypothesis about. And the second thing is about, do you think there is a role of delay in the circuit, or the role of dendritic dynamics, or not? What do you think about that? Sorry.
So for the first question on the pathological state, I think that this approach, which doesn't say anything about connectivity, in this case it was us controlling the correlated activity of the input. So connectivity from this point of view is left out. However, if you do the estimate of the transfer function, the way that we did it, one cell at the time, then you can take this transfer function and bring it very easily in a model where you have, say, physiological or pathological connectivity. What I think it might be interesting would be in the context of channelopathies, so those pathologies where ionic conductances, voltage-gated ionic conductances are either overexpressed or they have different kinetics. Those would alter in a way that it's maybe subtle the spiking patterns. So spikes will be still there, but maybe they will change. Say, I'm thinking of the some differences of some pathologies in which in some epilepsies, subunits of sodium voltage-gated channels are altered. So in that case, you have a different activation or inactivation of the sodium currents. And this directly reflects on the high Fourier frequency part of this filter. So we do have, not in the pathological context, but in the context of rodents versus humans having different subunits of sodium channels, the indication that you have lower or higher bandwidth. So having said that, you take that, assuming so that characterization of transfer of input-output signals, you can put them in. You can put it in computer sim in mean field like approach. So it could still be numerical, and there you can also add this pathological dimension of the connectivity, but that here is completely neglected. Is out of the. For the delay, yes, absolutely. I'm not an expert, but so this transfer function that I, in a way, try to sell you, because I show you, you see, if I use it, then I can predict quantitatively the data apart from a constant, from a proportionality constant. The transfer function is only a part of a more general picture, which comes from the so-called Fokker-Planck equation, the solution of Fokker-Planck equation, where you are estimating the probability of, for instance, finding the membrane potential at any given point at a given time, and by this you can say, okay, what would be the rate of emission of threshold crossing? And this can be done also for models that are not so simple, say not only leak, integrate, and fire. So there, colleagues, people like Maurizio Mattia and Paolo Del Giudice, they studied the presence of delays, and they matter. However, but here I'm not. I don't remember. I don't want to say on the intuition. I should be able to say something. Intuitively, the way I vaguely remember is that the presence of a delay was altering in the Fourier domain in the corresponding. So if it's a small delay, it maps to a very high Fourier frequency. But I'm not 100% sure that this sort of mnemonic trick saying, oh, if it's a short delay, it actually only matters at the very high Fourier domain features where the cell itself might be already attenuating strongly, and instead long delays would instead impinge on low Fourier frequency domain feature. I don't know whether this simplification holds always, but they are parts. They can change the dynamics. I'm presenting what operatively could be easiest to. So already Fokker-Planck. I need then afterwards to excuse to the god of physics for having pronounced or mathematician to having pronounced Fokker-Planck.
Anyone else?
Michele.
So we are. I would say we are right in time. It's 3 o'clock. So we were saying before that the seminars included with questions last around 1 hour, and so I think we can thank our speaker.